This is our energy and enzymes lecture. It provides basic background information for the cellular metabolism lectures we'll have next. Since metabolism is really just dealing with all of the energy transfers within the body, it's probably a good idea to start out with what's energy. Simply put, energy is defined as the capacity to do work. I think sometimes that's a little hard to visualize for some students. So we'll come back to what type of work that really is in a minute. For terrestrial ecosystems, meaning things on the surface of the earth, plants use energy from the sun to store energy in molecules. So photosynthesis allows plants to store energy because animals can't trap sunlight and are not capable of doing photosynthesis. We have to consume different biomolecules for energy. When we talked about biomolecules, we talked about how much energy you get from each one on average. So animals either need to eat plants or other things that ate plants. And that's an important component. So there are some ecosystems like deep sea ocean thermal vents and in caves and a few other places that don't use sunlight for energy because sunlight doesn't penetrate. So they do what's called chemosynthesis, and they use different chemicals to synthesize energy. And other than that, the ecosystem, to a large extent, from an energetic standpoint at least, is fairly similar. A couple other points about energy. The extraction of energy at best is about 40% efficient. We cannot completely burn energy, and what ends up happening is some of that inefficient burning comes off as heat, which is not bad for us because we're warm-blooded organisms and we need to maintain body temperature. And anytime we eat excess energy, that's going to be stored in biomolecules. And really the two big ones we'd worry about would be glycogen and fat. So energy is required for living processes. It's one of the components that distinguish living from non-living things. There's many others though. So when we talk about energy being the capacity to do work, there's three types of work that are important physiologically. One is chemical work. So chemical work, it takes energy to break bonds sometimes, and it takes energy to make bonds, certainly. Matter of fact, remember, a bond is just a favorable energy relationship. So in order to have a bond, you need favorable energy. So that's the chemical type of work. The next type is called transport work, and that's moving things across a membrane. Oftentimes against a gradient, and that requires a tremendous amount of energy over the day. More energy is spent doing transport work than the other two combined. And then the last one is mechanical work. And mechanical work is about physical movement, right? Not just your muscle moving, your heart beating, smooth muscle moving things in your, intestines, as an example, or even little molecular movement molecules within our cells called kinesin or dynein. All of those things are part of that mechanical work where it would require energy to do that. So the three basic types of work performed in living organisms: chemical work, transport work, and mechanical work. Now, the most important source of energy for every organism we've studied is ATP. ATP is adenosine triphosphate. It starts out as an adenine. Okay, so this is its nitrogen base. Remember, ATP is a nucleotide, and all nucleotides have a base, a sugar, and a phosphate. So adenine is our base here. Our sugar is ribose. It's a 5-carbon sugar. So that's ribose. So each one of these corners is a carbon, and then this one. And then we have a phosphate. Actually, we have 3 phosphates, so that's where the triphosphate comes from. Energy is released when we break this last bond. Okay, so since, again, bonds are favorable energy relationships, to make a bond, I have to put in energy. And if I break a bond, I can liberate energy. So this energy right here in this last phosphate molecule, when I break that bond, it liberates energy. When I do that, we have a phosphate. It's often called a P with a subscript I, or inorganic phosphate. And then an ADP molecule, right? So instead of having 3 phosphates, this one's gone, right? So we have 2 phosphates. So that's really important in terms of things. We don't have a whole lot of ATP stored as energy. It actually surprises students a little bit. And that's because we use so many molecules of ATP a day. If we stored all the ATP we needed for a 24-hour period, we would weigh about 350 to 400 pounds more, because that's how many pounds of ATP that we would have to store in our body to do that. Obviously not very efficient. So what we do is we store our energy more efficiently as biomolecules, and then convert those biomolecules to energy when we need it, for the most part. So, as I mentioned, we need to be able to transform energy from one form to another. There are 2 types of energy you can look at. There's a bunch of different ways to look at energy, to be honest. But one way is to look at kinetic energy versus potential energy. Okay? Kinetic energy refers to movement, right? Kines literally means movement. So kinetic energy is movement energy. And as an example, the person diving in is now undergoing kinetic energy, turning potential energy into kinetic energy. The person climbing is converting kinetic energy into potential energy. The reason why I said potential is potential energy is the energy of location. So the person at the diving board has a high potential energy in a gravitational field, right? The person in the water in a gravitational field has a low potential energy. It could also be potential energy, the energy of structure or storage. And all of those things are, examples of potential energy. So storage of fat really is potential energy, if you want to look at it that way. So that's kind of the 2 types of energy that we can look at in the human body, kinetic versus potential. And we're always converting one to the other and back and forth from things. Now, there are 2 basic types of reactions that we can talk about. One is called exergonic, and the other is called endergonic. Okay? And exergonic reactions release energy. And we go from a high energy state, that's what this purple line, I guess pink line is supposed to represent here, to lower energy products. And when we do that, we liberate energy. Okay? Endergonic reactions require energy. And here we see the reactants are a low energy state. We have to put in energy to get the reaction to go, and then with products that have a net higher energy. The reason why is because we put energy in. In the first one, the exergonic, the products have a lower energy, so we go from the high value here where it says reactants to the low value here when it says products, because some of that energy was given off during the reaction. So sometimes, physiologically, we couple reactions and try to make a favorable reaction happen to make a then unfavorable reaction happen. So we release energy in a favorable exergonic reaction and then use that energy in the next step in an endergonic reaction. And by doing this energetic coupling, we allow reactions to move. Now, the energy that it's actually talking about is called free energy. And we're going to define free energy as all the available energy stored in bonds in your body. Okay? So something that has a greater amount of bonds will have a greater amount of free energy. So, a really simple example would be a glucose molecule has less free energy than a glycogen molecule. And that's because a glycogen molecule, by definition, is made up of multiple glucose molecules. So free energy is the energy that is stored in bonds. So here's another way to kind of look at those favorable and unfavorable energetic reactions happening. So this kind of looks at what's sometimes called the ATP cycle. So we'll start up here with ATP. And what happens is you put a little water, okay? And when you do that, you split the ATP into ADP plus that PI I mentioned, that inorganic phosphate, and you give off work. And then we can take this ADP and inorganic phosphate, combine them back to make ATP, but we have to put in energy to do that. Okay? So this side, this left side, we get from what's called catabolism. Catabolism is breaking down. They tend to be energy releasing processes. And so when we break down our food for energy, that's called catabolism. It's a catabolic process. So we just go through this ATP, ADP cycle all day long. And we really only have, we'll talk about at the end of the metabolism section, but we only have enough ATP for literally a couple seconds of exercise, and that's it. And if we don't regenerate the ATP, then we'll run out of it very quickly. Obviously, most of us can exercise more than a couple seconds, so we are regenerating that ATP. So we use these reactions to drive unfavorable reactions and have things happen in our body that makes things work better. Now, the energy required to make a reaction go is called the activation energy. Okay? Without getting too technical, okay, the activation energy provides enough energy to reach what's called the transition state. And the transition state is the highest energy needed to change the bond configuration of molecules, so to make and break bonds. Okay? Some reactions have a high transition state, which means we have a high amount of activation energy needed, and some reactions have a very low energy of activation that's needed. And really, it's the ability to make these reactions go faster that allows humans to physiologically work the way we do. And so without being able to lower the activation energy, we wouldn't be able to function. So as an example, if we take sucrose, right? So sucrose is table sugar. Okay, so if you took sugar and put it in coffee or tea, right? That's sucrose. And if you took sucrose and you let it do its thing, eventually it'll break up and do its two components. So sucrose is a disaccharide, and sucrose's two components are glucose and fructose. Okay? If you let sucrose break down all by itself, it would usually take a number of years for sucrose to break down to glucose and fructose, right? So years. However, if we use an enzyme, in this case, you can see it here in the bottom middle, sucrase, right? That conversion happens literally in seconds. So the way enzymes work is they're called a catalyst. A catalyst is something that accelerates or speeds up a reaction. So it makes the reaction rate go faster. Catalysts are regenerated and reused. There's not a 1-to-1 ratio. A catalyst can be used again and again and again. So that's a good example of what an enzyme is. And the way they actually function is they lower the activation energy for a specific reaction. So this black line follows the energy needed to reach transition state and then go back down to products. Well, the red line shows the same reaction, but this time with an enzyme that reduces the activation energy. So you can see that it's easier to do that. It'd be sort of like, not a bad analogy is a remote control for your TV is sort of like an enzyme. Okay? If you had to physically get up, walk to the TV, and change the channel, and then go back down and sit and watch TV, you're going to change it a lot less than you would if you had a remote control. And the reason is it takes so much activation energy. You have to stand up every time when you want to change the channel. But if you have a remote control, all you have to do is, pick up the remote control and move your thumb, right? So you don't need as much activation energy. Therefore, you're more likely to change the channel and look at a different channel. So enzymes are catalysts. They accelerate reactions. They're regenerated and reused multiple times, and they work by decreasing the activation energy. Now, all reactions are technically reversible, but typically reversible reaction can proceed in both directions, while irreversible reactions favor one direction. So as an example, if you look at this graph, okay, here is what we're trying to do is we're trying to turn two things, A and B, into two other things, C and D. And if you look at this activation energy needed, it's a tremendous amount of, sorry, let me go back. It's a tremendous amount of energy to reach this transition state, okay, and then go down to C to D. If you wanted to go backwards, right, you'd start at this energy level and you'd have to have input significantly more activation energy to reach transition state before you went back. So this is called an irreversible reaction, which is technically not correct, but realistically it is, because there's very few cases where you're going to have enough energy to go backwards, right? There's cases where you can go forwards, and that's probably, if you're looking at a chemical reaction in the human body, that's what we're designed to do. But going backwards is virtually impossible because we just don't have enough energy to overcome the activation energy. If you look at a reversible reaction, you'll see that in the reversible reaction, there's not nearly as much activation energy required. And so these reactions are much more likely to go. And then if you go backward, you can see you even need less activation energy to go backwards. And so that's much more likely to happen than something over on this side. Now, reversible reactions obey what's called the law of mass action, or sometimes known as La Chatillier's principle. Basically, if you have a reaction in equilibrium, so let's say you have A and B and then C and D, all right? And if you start to make more A and B, right? If you start to make more A and B and it's a reversible reaction, what your body's going to do by mass action, it's going to drive the equation to the right. We're going to go towards the right side. So as we move things over to the other side, eventually they reach equilibrium again, but now they're equal on both sides. So mass action says the more you have on one side of a reversible reaction, the greater amount of product you're going to have on the other side. So reversible reactions restore equilibrium. So if we add more reactant, we'll make more product. If we remove product, we'll make more product. If we remove reactants, we'll make more reactant by going backwards. So wherever the smallest amount is, that's the direction we're going to go in a reversible reaction. One last thing about reversible reactions before we move on. Reversible reactions are good because they help us control things. But there's a cost at that, and we see this a lot in physiology. There is a cost benefit approach that you have to look at. So we get more control of a process by having a reversible reaction, because that way we have an enzyme going from A to B, let's say, and another enzyme, a completely different enzyme, going from B to C backwards. And by being able to control those independently, we can control what the speed of the reaction is happening, and it gives us much more control physiologically. So we have certain reactions that can be better controlled by going in both directions. And to be honest, those reactions that have even more control have different enzymes that control each direction. So you have an enzyme that goes from A to B, and you have a different enzyme that goes from B to A. We have many reactions in the body that are reversible that have only one enzyme in either direction. So by having two enzymes gives us more control as well. Now, when we're talking about this section and then the one that follows for the next exam, we're talking about metabolism. And metabolism is the sum of all the chemical reactions in an organism. And all of these, don't look at them in a lot of detail. The whole point is these are all metabolic processes, and these aren't all even close to all of them, but it just shows you how complex this whole thing can be. Now, when we look at metabolic processes, right, all the chemical reactions in the body, really we can divide it into two basic ones. Number one, what we call anabolic. Anabolic are also known as biosynthetic. Okay, I'll come back to those ideas in a second. And then catabolic. Catabolic are degradative. Those are breaking down. So I think it's easy for people to remember anabolic because generally speaking, most of you have heard of anabolic steroids. And most people also know the general effect of anabolic steroids would be somebody to have bigger muscles, right? So if you think about anabolic, it means to make larger molecules, right? Now, anabolic processes consume energy, right? Because if I need to make a bigger molecule, I need to take a small molecule and put more bonds on it. Remember, bonds are energy, so I have to put energy into that. The opposite of that is catabolic. Again, those are degradative pathways. They're breaking things down. So we go from a large molecule into smaller molecules. When I do that, I break bonds. And when I break bonds, that releases energy. So catabolism is what we do to our food, whether it's carbohydrate or lipid primarily, so that we can liberate energy. Now, what happens for this process is that enzymes speed up these chemical reactions in our body, so it can happen at a much faster rate. We're going to talk about a process called glycolysis in the next section. And glycolysis is a very important energetic pathway where we break glucose down into pyruvic acid. It's a fairly simple process and involves 11 steps. Every step is catalyzed by at least one enzyme. Beginning of the pathway and the end of the pathway, we have independent enzymes for each direction, while in the middle of the pathway, we have the same enzyme that goes in either direction. But it's enzymes that allow these chemical reactions to happen. If we didn't have enzymes in the human body, it's estimated that it would take that process we're going to talk about later, glycolysis, take about 20 years in the average human to completely break down a glucose molecule. Yet in our body, under especially very high energy need events, like if you're sprinting to your car for some reason, then glycolysis takes place literally in less than a second. So it's very, very, very fast. And that's, again, the use of enzymes that really helps that appreciably. So enzymes speed up reactions by lowering the activation energy. So we call them a catalyst, right? And one of the things about enzymes is they exhibit specificity. Now, when we talked about transporters for both passive and active transport, we said, well, most of the transporters are protein transporters. And as a protein transporter, it's going to have three basic attributes. One, it's going to exhibit specificity. Only specific things bind to that. It has to have the right three-dimensional shape. It exhibits competition where different ligands, remember a ligand is just something that binds to a receptor, different ligands compete for binding sites, right? And the one that either has the highest concentration or the highest affinity, remember affinity is attraction, then that's going to bind the best. And then also those receptors, because there's a finite number of them, exhibit saturation. And as you increase the amount of, if you're talking about enzyme-substrate, but if you're talking about other things, it could be, just not product of a substance, then you're going to increase the reaction rate up until a certain point, and that point would be when we reach saturation. So enzymes and protein receptors can be saturated, which means adding more of the substrate won't do any good because all the binding sites are already active. You can't go any faster than that. So these enzymes show this specificity that's very important. The reason why they do that, and the easiest way to think about it, is enzymes, for our purposes, most enzymes are proteins. And proteins, when a substrate binds, is the exact same concept as the specificity, competition, saturation of a protein transporter for, active transport or facilitated diffusion for that matter. So we can change enzyme activity in a number of ways, and we call that modulated. Okay, so enzyme activity is modulated, changed in a number of ways. One is physical factors. So temperature and pH can change enzyme activity, speed it up or decrease it depending on what's going on. Sequence modification. We have what is called proteolytic activation. So again, most enzymes are proteins, and when they first get released, a number of those are fairly inactive. And it's not until we chop off a section of the protein where it becomes more active. We also have isozymes. Iso means the same, zyme refers to enzyme. So we have the same enzyme in different tissue that work a little bit differently. So as an example, there's an enzyme called lactate dehydrogenase that is different in the heart than it is in skeletal muscle. There's also another one called phosphofructose kinase as an example, which is different in different muscle tissues as well. So those are called isozymes. Same enzyme, different tissue. But it's not exactly the same probably. It's very similar, but not exactly the same. We can also chemically modify an enzyme or a substrate. There's two ways to do it. One is called allosteric, where you bind to someplace else on the molecule, and the other is called covalent, where you bind something covalently to the molecule. And then we can also affect activity with the concentration. So if we increase the substrate, we increase the activity as long as we haven't reached saturation. If we increase the enzyme, we provide more binding sites, so that should increase the reaction rate. And we can decrease things by competitors. So we can have competitors that, if, for example, a competitive inhibitor would block the active site and prevent the enzyme from working. So let's kind of go through those a little bit more detail. So these two drawings right here are supposed to represent two different enzymes. Okay, and these ligands, right, are the substrates. And if you look, right, you can see that enzymes have specificity. So this fits here, the A fits here, and the B fits here. So any of these three substrates, also known as ligands in general. Anything that binds to a protein receptor is called a ligand. Some people say ligand. Right? So that's still specificity, because this thing right here down, this green thing down here, wouldn't bind because it's not the right shape. It's similar, but it's not the exact right shape. Well, when we look at protein Y, only the C will fit, and the A and the B will not. And the reason why is they have these long arms that will get in the way of these little things pointing up. So you have to have the right three-dimensional shape in order to bind to the enzyme. And it's really just the three-dimensional shape. What will help binding are two things. One is the affinity. The affinity is the attraction of the substrate in the enzyme. So if you look at the bottom one, you'll see that the first figure on the left bottom says protein, and it's got a little positive charge in the middle of the active site. And it lines up perfectly with the negative, right? So the negative charge on the ligand. Okay, so that, again, the ligand is just your substrate, if you want to call it the substrate, the thing that's going to bind to the enzyme and have the reaction. So this has a high affinity, a high attraction, because the opposite charges attract. If you look at the second one, right, there's no plus anymore. Remember, the plus was there, now it's gone. So this has an intermediate affinity. That, yeah, there's a negative charge here, there's no corresponding positive charge there, but it can still bind because it has the right shape, it has the right specificity, and there's nothing to oppose the negative charge on the ligand. If you look at protein number 3, though, right, you see a low affinity, and let's say they don't have it, but let's say this was negative right here, right? The negative charges wouldn't connect to each other very well. You have this low affinity binding site, which means that reaction rate would be really, really slow, relatively speaking. Okay. The other thing that matters is concentration. So if we increase the concentration, we're going to increase the activity, again, up to saturation. So if you look here, right, as we increase the substrate concentration, higher, 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 I don't want to push that, higher, 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 higher. When we reach a saturation point, notice it goes straight across. Okay? And what happens at saturation, it basically says that all of the binding sites are full, and since they are, we can't go any faster because they're all full already. And you can't go any faster than full. Now you could change that if you added more enzyme, because now you'd have more active sites, but if you have the same amount of enzyme, then you're going to reach a saturation point. Okay? And that's the maximum reaction rate for that enzyme. All right, so enzyme activity can be modulated by environmental conditions. Now, most people don't teach it this way, but I think really it is true that enzymes have an optimal temperature. Okay? They have a temperature where they tend to work best. Okay? And so some enzymes work really well at one temperature, while other enzymes work really well at another. So here's an example of an optimal temperature for the human. And if you look at this black line, it reaches the peak activity at about 37 degrees Celsius. Why is that important? Well, the body temperature is 37 degrees Celsius. But here is what's called a thermophile, a archaebacteria that resides in these sulfur ponds, these hot sulfur ponds, sometimes at Yosemite. Not Yosemite, sound like the president, at Yellowstone, right? So at Yellowstone National Park, they have these pools of basically bacteria. And because these pools are from hot springs, they're very, very hot. And so because these bacteria live in this environment, their optimal temperature is close to 80 degrees Celsius, right? So almost boiling, right? 100 degrees would be boiling. So almost boiling point there. And they work best at that high temperature because that's the environment they're in. And if we moved away from the temperature in either case, right, from that optimal temperature, activity would go down, all right? pH is the same way. We have optimal pHs, the optimal pH for that enzyme to work. Here's a good example of pepsin. Pepsin, pep refers to stomach. Pepsin's an enzyme we have in our stomach. And notice its optimal pH is 2. And then here's the optimal pH for an enzyme called trypsin. Both are what we call proteases. They break down proteins. Okay? But trypsin, it's an intestinal enzyme, and its optimal pH is 8. Why? Well, when you eat, you release stomach acid, and the contents of the stomach after eating have a pH pretty close to 2. So it makes sense that an enzyme in that environment would have a pH pretty close to 2 for its optimum. When we look at trypsin, trypsin has a pH that is 8. Right? That's 1,000,000 times different in the amount of hydrogen ion concentration that works best for that enzyme. So the intestinal enzyme is in the intestine, which makes sense. And intestines, after someone eats and food moves through the system, have a pH probably closer to 8 because of all the bicarbonate released. So the pH goes way up after it leaves the stomach, and the enzymes that reside there have an optimum pH at a much higher level. So, here's an example of, pepsin activation. Pepsin is actually released as a enzyme that's not quite complete. Okay, and that's called pepsinogen. When you see the word ogin, and sometimes we use the word pro in front instead. When you see the word ogin, it generally means it's an inactive form. So release this proteolytic enzyme, pepsinogen, right, from our chief cells into into our gut, into our stomach. And what happens is the HCl activates the pepsinogen into pepsin, and then pepsin activates more pepsin into pepsin. But the key component is that we're cleaving off a bit of the protein, and that's that proteolytic part we talked about before, where sometimes they're inactive and they can be activated by other sources. We can also alter activity by looking at chemical modification of enzymes. There's always the possibility an enzyme needs a cofactor or coenzyme to work. A common coenzyme are vitamins, where you have to have a vitamin couple with the reaction in order to see it take place. We're not going to worry about those so much, but we're going to worry about allosteric and covalent modulators. So an allosteric modulator, allos literally means somewhere else, works somewhere else other than the active site. So here they call it the binding site, right? And that's where the ligand or substrate binds to the enzyme. That's what they call the active protein. Okay. So allosteric, so this is the binding site. The allosteric part is when, and that's what this A stands for down here, binds someplace else on the molecule. So it's not in the active site. Okay. And ATP and ADP work really well doing that. When you're making ATP through metabolic processes and you have sufficient ATP, then you really don't need to make a lot of ATP anymore. And what happens is you inhibit that reaction and it slows down the ATP production. And the way it does that is it binds to an enzyme allosterically and then changes the active site a little bit so that the activity slows down. Okay? ADP does the opposite. Okay? So it's actually identified here as an activator. If you have a lot of metabolism going on, as we saw in an earlier slide, that you're going to take ATP and make a lot of ADP, right? So you're going to take the energy from ATP by cleaving off that last phosphate, and when you do, you have ADP. But when ADP starts to build up, it's saying that you don't have as much ATP as you need, because there's a what they call, like I said before, the ATP-ADP cycle. And high ATP means slow everything down because we've got enough energy. And low ATP means speed everything up because we need to make more energy. So by binding ATP or ADP allosterically to different enzymes, we can speed up or slow down the reaction rate and help control what it's supposed to do better. Covalent modulator is when we bind covalently some other molecule to a protein, again, to alter activity. Very common thing to do is with phosphate, okay? And this is showing, there's a couple things that, kind of show how this works. But if you bind phosphate to this part of the enzyme here, so the little brown thing is the enzyme, it shows the enzyme going from here, this shape, where it's not going to fit the ligand, right, to this more open space after you bind phosphate, right? So that's allosteric. Oftentimes this phosphate, because it's always going to have that -2 charge there, will increase or decrease the affinity of the enzyme and substrate. You can speed up or slow down a reaction there. Allosteric, this again shows the allosteric modulator molecule binding someplace else. The active site's here with the arrow, right? And in this case, it's an activator, right? Because once we bind the purple circle there, this goes from a triangle to a rectangle, so we can easily slide the ligand in and have the reaction take place. Now, you can have an allosteric modulator inhibit the functional site or binding site or active site, whatever you want to call it. So this, again, purple lavender molecule binds, and we go back this way, and all of a sudden that changes the active site. We're going to slow down the reaction because this doesn't fit in this three-dimensional shape. So that would be called a non-competitive inhibitor. It's an inhibitor because it slows down the reaction. It's non-competitive because it's not in the active site. Okay? But if you put something in the active site, right? So here's a normal reaction. But in competitive inhibition, we put an inhibitor here in the active site, and now we cannot bind the substrate because it's physically in the way. Not a bad analogy is that, let's say you went to school and you came at 9:28 for a 9:30 class. Well, if you've ever been on campus to DVC at that time, you'll know there's virtually no place to park. Okay? And the reason why is there's somebody in each stall. Each one of those cars in each stall in the parking lot is a competitive inhibitor. It's in your way physically. A number of years ago when they put the solar power panels in the parking lot by our building, they did it in the summer. And what they did is they basically closed off the entire parking lot and roped it off and then started to install the panels. In this case, it's a non-competitive inhibitor, right? You still can't park, but it's not because something's in your way of the parking spot. It's because you can't get in the first place. So that's like not a bad way to think of a non-competitive inhibitor. So whether it's allosteric or covalent, right? In terms of the modulator, they can increase or decrease activity, right? If it's an inhibitor, by definition, it's going to do what? Decrease activity. So we use a couple of these concepts to talk about how do we control metabolism. One way to control metabolism is to use separate enzymes for reversible reactions. So here's an example of an enzyme that's really fast. We talked about this already in Lab 8.4 discussion, where you take CO2 and water. With an enzyme called carbonic anhydrase, it forms carbonic acid. This is the fastest known enzyme in the human body. It will catalyze up to 100,000 of these reactions in 1 second. Okay? And if you have a lot of CO2 and water, you make a lot of carbonic acid. That's that mass action we talked about before. If you have a lot of carbonic acid, it'll make a lot of CO2 and water. We don't have as much control over this one. Here's an example of kind of a similar thing that's done a little differently, though. And if we take glucose and phosphate, that's what the PO4 is, right? We put it on the glucose, on actually molecule number 6, the 6 carbon, it becomes glucose 6-phosphate. And what's interesting about this reaction is from going from glucose to glucose 6-phosphate, there's a specific enzyme called hexokinase, right? So it only goes in that direction. And then we have an enzyme going the opposite direction, glucose 6-phosphatase, that will turn glucose 6-phosphate into glucose. By having separate enzymes, it does cause us to make more material, because we have to make twice as many enzymes, right? It does increase the possible chance of errors. The more enzymes you have to produce, the more chance you're going to have some sort of error in that production. But the bottom line is it gives you a tremendous amount of control. And so let's say you needed energy. This is the first step in glycolysis, where we take glucose and break it down into energy. Well, if I need energy, I need to activate the hexokinase, right? I need to make this enzyme over here. So here's my enzyme here. I need to make this enzyme. I need to activate it. I need to make it more pluses. And the reason why is I want to turn glucose, and I want to go this way as fast as I can into glucose 6-phosphate so I can make as much energy as I can. I do not want to go in this direction. I want to inhibit this enzyme over here. And that's because I want to force glucose to go to glucose 6-phosphate and then go and make me a bunch of energy. If I am just converting glucose into glucose 6-phosphate and then just as fast converting glucose 6-phosphate back into glucose, that's not going to do me any good. So by having these separate enzymes, I can differentially control metabolism. And again, when we went over those 4 basic themes of physiology, I said, control is one of the ones I would add to that list as very important physiologically. So that's the first thing. So another thing that regulates the metabolic pathway is controlling the enzyme concentration. As a matter of fact, there are many historical studies in physiology that looked at exercise training and the effect on enzyme concentration. And that's really one of the reasons why you can run faster or longer when you start training, is you have more enzymes in your mitochondria and cells to make more energy faster, more efficiently. So controlling enzyme concentration is important. Modulating enzyme activity, we just talked about that, by looking at concentration or affinity or competitive or non-competitive inhibition, allosteric or covalent modulation. All of those things can change the enzyme activity, speed it up or slow it down. Our body maintains the optimum ratio of ATP to ADP. And again, as you start to make too much ADP, Your body says, oh, we need more energy, and it speeds up your metabolic processes. If you make too much ATP, your body says, oh, I've got enough ATP. I don't need to start sending it off, whatever. So that will slow the metabolism down. I mentioned this already, using different enzymes to catalyze reversible reactions. It gives us more control. And then isolating enzymes within organelles. So I think most of you understand that the mitochondria is an important organism for energy. And the mitochondria is a membrane-bound organelle with an inner membrane that's all folded called the cristae. So by being able to isolate enzymes within certain organelles, it allows things to take place in a more orderly fashion and allows things to happen the way they do a little better. So this is just a summary of some of the things we talked about so far. Metabolism is all the chemical reactions in the body. When we have a chemical reaction, we can make or break a bond. Anabolic processes make bonds. They make bigger molecules. They consume energy. Catabolic processes break bonds. They release energy. The free energy, or some people might know it as the Gibbs free energy, is the potential energy stored in bonds. The more bonds you have, the more potential energy you have. The less bonds you have, the less potential energy you have. When we look at metabolism, metabolism is organized into different pathways. There's a number of different types of pathways you can have. Sometimes they're linear, right? And A goes to B, and B goes to C, and C goes to D. When we look at that, right, that A is often, whatever A is called the substrate. Right? So the substrate. The end product would be D. End product's probably a better name than just product. So D would be end product. B and C would just be intermediates because they're in the middle, right? And enzyme 1 catalyzes the reaction from A to B, and enzyme 2 from B to C, and enzyme 3 from C to D. Not all metabolic pathways are linear. They can be circular, like the Krebs cycle. All right, and basically we start with the reactant. It gets moved to an intermediate. It gets moved to another type of intermediate based on what we're doing in class. And then we split out a product. So you spit out the product, and what's left of the intermediate can be used with the reactant to, again, get a different intermediate. So intermediates are things found in the middle of metabolic pathways that help in terms of understanding what's going on inside of them. So you can also regulate these pathways through a few other methods. One is an extracellular signal, like a hormone. So you can increase or decrease this activity with a hormone, or you can actually use the pathway itself to control, and that's called feedback inhibition. Let's say we're going through this process where we're going from A to B and C and D and E and so on and so on. And eventually we get to Z, and Z is our desired product. Well, we don't want to keep on producing Z over and over and over again and not waste Z if we can help it. So what happens is when we start to get a lot of Z accumulate, then Z will feed back to A and slow down the reactions. Okay? That's called feedback inhibition. Ultimately, the most potent regulator is ATP-ADP ratio. Another way that we help control metabolism is we compartmentalize, or compartmentation, and that adds an additional level of control. So as an example, breaking down glucose for energy, there's 3 basic steps we have to do. We have to do glycolysis, and then we have an end product of pyruvate. Pyruvate goes into the mitochondria, and then we go through the citric acid cycle there. And then eventually we use the electron transport chain as well to help make energy. So By having things divided and specialized, we're much more efficient in terms of the energy that we make. So that concludes the enzyme presentation and this section.